Good day everyone, this is Myla Dimihia, your teacher for General Biology 2. Today we are going to discuss the central dogma of molecular biology. The central dogma of molecular biology deals with a detailed residue-by-residue transfer of sequential information. It states that such information cannot be transferred from protein to either protein or nucleic acid. The central dogma of molecular biology can be summarized in the following. So DNA, which will be transcripted into RNA, then translated into protein. This is also another illustration of how the central dogma of molecular biology happens. The central dogma of molecular biology starts with DNA replication. Each strand of the double helix serves as a template of the synthesis of a new strand. So there are proteins which are needed and enzymes which are needed in DNA replication. So we have the following: initiator proteins, helicase, SSBP, gyrase, primase, DNA polymerase 1, DNA polymerase 3, ligase. We'll come to know each of these later on as we go on with our discussion. The mechanism of DNA replication. It starts with initiation. Initiation states that this is the preparation or preparing the double helix for the complementary base pairing. So the different proteins and enzymes which are needed here are initiator protein, helicase, single-stranded binding protein, and a gyrase. So this is one way of how we are going to illustrate replication. Another, so the mechanism of DNA replication. Next, we have also elongation, connecting the correct sequence of nucleotide into a continuous new strand of DNA. And the needed proteins and enzymes are primase, DNA polymerase 3, DNA polymerase 1, and ligase. Again, another mechanism of DNA replication. So we have the summary. So for the summary of DNA replication, it states that it starts with unwounding of the double helix. So after which you will identify the different ways and means of how these different structures will bind, and at the same time will also carry the different kinds of protein and enzymes which are needed in order to unwound the DNA. Now in the replication of prokaryotes and the eukaryotes, so we have the differences. For prokaryotes, so when say prokaryotes, these are the different organisms which do not have a true nucleus. Unlike with the eukaryotes, they have true nucleus. So replication of DNA, of circular DNA, begins in a single origin and proceeds bidirectionally under the circle or around the circle that uses DNA polymerase 1 and DNA polymerase 3. While on the other hand, eukaryotes, replication of linear DNA molecule is initiated at the numerous origins along the DNA. And it uses the DNA polymerase, the gamma, sigma, and the beta. Next is transcription. It is a synthesis of an RNA molecule whose base sequence is complementary to the base sequence of the template DNA strand. The basic processes in RNA synthesis are binding of polymerase to a promoter sequence, initiation of RNA synthesis, elongation of RNA chain, and the termination of RNA synthesis. RNA polymerases are the RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. These are all included in your module. For the initiation of transcription, an RNA, which stands for ribonucleic acid polymerase, attaches itself to a specific sequence in the DNA called the promoter sequence or the initiation point with the help of polypeptides called the sigma factor. So as you can see, the yellow structure is referring to that of the sigma factor. Elongation of the transcript. The RNA polymerase moves along one of the strands of the DNA called the anticoding strand, separating the two strands as it goes along and complementing DNA nucleotides with RNA nucleotides. As soon as the sigma is transcribed, the DNA strands re-anneal because DNA-DNA interaction is stronger than DNA-RNA interaction. A single-stranded RNA is thus produced. This continues until the enzyme reaches the termination point. And how will we know if the termination point has already been reached? So we'll come to know that later. Now, for the termination of transcription, another protein factor called the Rho binds to the enzymes and dissociates it from the DNA to prevent further transcription. This produces an RNA complementary to the anti-coding strands, and this could give rise to three types of RNA, namely the mRNA, the tRNA, and the rRNA. M for mRNA stands for messenger, while T, that's the transfer, and the R is ribosomal. Why ribosomal? Because the ribosomes are attached to that of the RNA. Now, what are the different functions of the types of RNA produced by the transcription? mRNA provides the template for the amino acid sequence of protein. tRNA, this transfers amino acids from different translocation in the cytoplasm. And rRNA, this will be used for binding ribosomes, machinery for synthesizing proteins in the translating mRNA. So as we can see, the red structure there, which is just like a hoop of the hairpin, this will indicate that termination has already been done. Okay, so RNA processing after transcription, what are needed here? Adding of methylate cap or the poly A tail, deleting introns from a primary transcript, and determining which exons from a primary transcript are joined together to form a mature RNA and joining exons of different genes. So structures here are the RNA splicing, the amino acids. Now, for the translation, the RNA processing, the process in which RNA or mRNA transcript is used in the synthesis of protein. Now, these are the different steps of translation process. You have the initiation. A ribosomal subunit binds to a molecule of mRNA. In prokaryotic cell, the mRNA binding the site on the subunit recognizes a specific nucleotide sequence in the mRNA just upstream in the start codon. An initiator tRNA with anticodon UAC pairs with that start codon AUG, which carries the amino acid. Yeah. And then the translation initiation complex. The arrival of the large ribosomal subunit completes the initiation complex. Proteins called initiation factors are required to bring all the translation components together. The initiation tRNA is in the P site and the A site is available for the tRNA bearing the next tRNA bearing the next amino acid. Translocation. The tRNA in the A site is translocated to P site in taking the mRNA along with it. The tRNA in the P site moves to the E site and is released from the ribosome. Now, for the termination of translation, elongation continues until stop codon in the mRNA reaches A site of the ribosome. A protein called the release factor binds to the stop codon in A site. The RF causes the addition of water nucleotide instead of amino acid to the peptide chain. Now, the differences in the translation of prokaryotes and eukaryotes are there. So in terms of ribosomes, initiation proteins, initiation, and elongation. So this ends now our lesson on the central dogma of molecular biology.